Okay, so I'm going to just dive in and tell you a bit about the Freshwater Biodiversity Information System. Essentially, it's a collaborative project with a number of organizations led by the Freshwater Research Center and with our lead partners, Kartoza. Just to reiterate the fact that it's, a number of people were involved and remain involved in this project, from the main group of people based in Cape Town to the group in Tim, who's based in Portugal, and our lead developer, Dimas, in Kartoza, who lives in Indonesia. Okay, so the journey so far, just going to close this because it keeps moving. The project essentially was funded by the JRS Biodiversity Foundation, and then co-funding came in from our local South African National Biodiversity Organization Institute. And the plan was to develop an information system for evaluating long-term change in South African rivers. We started the project with a planning grant, which kicked off in August, and it was roughly 9 months of planning. And during that time, we held a planning workshop with all of the potential stakeholders and end users of FBIS, trying to gain insight into what their needs were. And it resulted in the development of our technical proposal, which was submitted in May. Luckily, everything went well, and we got the funding for the technical grant, which kicked off in November 2017 and finished recently in December 2020. The first thing we really did with our technical grant was to hold a very intense and extremely useful user requirements workshop in January. Essentially invited all of our key stakeholders and end users to try and figure out what their requirements were right at the get-go, and this strongly informed the design and functionality of FBIS. We also developed the system iteratively with several workshops and 3 releases, with the last one being in July 2020, which is essentially the one that we're going to demonstrate later on today. So in terms of our initial aims, the plan was to mobilize, unite, and provide end users with access to freshwater biodiversity data from a range of taxa and taxonomic levels, and to facilitate the inflow of current as well as future data from these data sources. And in this way, allow for sort of long-term assessments of trends in not only river biodiversity, but also in health and indice data based on the biodiversity data. And obviously the end goal with all of most information systems is to guide river management decisions, and also in our case to also inform strategic long-term monitoring efforts. So briefly, the objectives were obviously to design our information system, and that's really what we'll be focusing on today. Mobilize and importing the baseline biodiversity data, identifying sites, especially using FBIS to inform where long-term monitoring can happen or should happen, and very importantly, to train our key organizations in how to use the information system. So briefly, the components of the first phase of FBIS were to mobilize biodiversity data. So you can imagine when you first start developing a system like FBIS, everyone has their pet taxa or their pet needs, and the overall wants and needs of the user community are very, very broad. But it's quite impractical to tackle everything in one go. And so we decided to prioritize the first phase to include rivers data, and specifically algae, inverts, and fish. We also included all the physicochemical or abiotic data associated with the biological data, as well as mobilizing what we call GIS contextual and aggregating layers. And essentially these became the spatial filters for querying data or selecting a subset of data, and for layers for visualizing the data. And Jeremy will obviously unpack this in more detail when he goes through the live demo. And then we also hooked onto a number of third-party databases, specifically GBIF, which forms the taxonomic backbone of FBIS. And we harvest data from GBIF, which includes research-grade iNaturalist data. We also link to IUCN for all the information on conservation status of each species, where it's available. And then we link to two very important other third-party databases. The first is a citizen science database called MiniSAS on river health. And the second is through a collaborative project award, we link to the Department of Water and Sanitation's water quality data. And very importantly, we have in the back end a knowledge database, because obviously users always want to know where the data comes from. And this is essentially PDFs, DOIs, etc. So the metadata about the data itself. Excitedly, we did get funding for a phase 2, and I'm very briefly just going to mention where we're going on the next 3-year journey. It hasn't kicked off yet. We're still waiting for the contract signing, but hopefully by April. So we will be adding new modules and mobilizing new biological data, specifically a neuron data, developing a thermal module, which speaks to some climate change work we've been doing, and expanding the rivers to include inland wetlands, and again, including data for algae, inverts, and fish, and then adding in wetland and riparian plants. Also, we'll be taking parts of FBIS and developing a mobile app. And very importantly, this phase aims to integrate the outputs of FBIS, so the data, the reporting, etc., and almost hardwire it into freshwater decision-making frameworks and pipelines in South Africa. And a lot of the reasoning for this is not only to make the data more useful, but also to speak to the sustainability of the system. Our thinking was, if FBIS becomes an indispensable tool in speaking to, for example, EIAs and the National Biodiversity Assessments, then, and the IUCN status assessments, if FBIS becomes integral to making these decisions, then it'll be more likely to be sustained, because most people who've developed information systems know that securing long-term funding once the development has happened is always a challenge. And so we're trying to address this head-on in the new project. We also plan to expand the data coverage, mostly through training of data providers. So expand the geographic framework and train the number of people who use FBIS and contribute data to FBIS. Okay, I did want to now just speak to a little bit about the partnerships and emphasize the fact that we really 100% believe in the partnerships that we've created and emphasize the importance of these partnerships. We've been lucky that a lot of organizations have been excited by FBIS and seen the potential value in it. And essentially, we had a good turnout at our first workshop, and then they've been part of the entire journey. So what we did was really take the time to understand what a user needs and use this to inform both the design and the functionality of the system. And we did this in a workshop scenario where we had a number of personas, from policy advisors to resource managers, conservation, biodiversity, researchers, and consultants, and essentially asked these individuals to put on a hat of one of these personas and then tell us what their needs were in terms of what are they intending to do with the data. So what are the reporting requirements? For example, biodiversity or river health assessments, fish atlases, scientific papers, EIAs, as I mentioned. We asked specifically what the outputs were that they needed. So drill down into the details of the type of maps and tables and figures. They were interested in trend analysis. Most individuals wanted access to the raw CSV files for further data analysis, and then data interpretation tools where this was possible. We asked about the scale of the reporting needs, and essentially this came down to national, regional, as well as very local scale reporting. So we had to develop a system that accommodated all of these. Then whilst we had a lot of individuals in the same space, we unpacked a bit more what data are available. Obviously, we already had a good idea of what these were as the project team, but we asked how accessible the data are. What is the format of the data? And what are the priorities for data inclusion? Again, realizing that we can't do everything in one go. And one of the things that came out really strongly that at all of our interactions with our stakeholders and end users was that quantity must not compromise quality, and that the value of high quality, reliable data is really, really key. Better data obviously equals better decisions, and we strive to make FBIS a source of information rather than a data sink. Okay, so I'm going to now just provide a couple of screenshots introducing you to the Freshwater Biodiversity Information System before Jeremy does a live demo. So essentially it's for tracking change in South Africa's freshwater biodiversity. At the moment, we've got 4 modules: fish, invertebrates, and algae, and very recently odonate adults. This is through a collaborative JRS project that's also with FRC, and very excited to be able to serve these adult odonate data as well. So the system we've designed, we've tried to make it as user-friendly and intuitive as possible. So the first thing we did was to make, develop the spatial interface and a spatial front end that you can use to navigate around the system. We've included a number of filters for selecting a subset of data that you may be specifically interested in. We've provided a number of layers which essentially allow you to visualize the data with a lot of different backdrops depending on your specific interests. And then we provided a number of base maps varying from sort of satellite-based maps to topographic-based maps to terrain, which can be interchanged depending on the needs of the user. We've developed functionality for adding a site, as well as adding data. And in this case, the example I've used is South African scoring system or river health indice data, where we've made the data capture form essentially mimic the field data sheet that users are familiar with to make the whole process of capturing data as easy as possible. We've also got other aspects like adding fish data, so individual fish records. And I want to just mention at this stage that in FBIS, although the focus initially was on serving, occurrence data like the presence of a specific species, in developing the system, we realized that for a little bit of extra effort, we could include not only information about the biotopes or habitats of where this fish occurred, but also the sampling method used, the sampling effort, and in this way quantify not only presences but abundances of the biodiversity data. Obviously, if a user doesn't have this information, it just becomes species X wasn't present at this site at this date. But if this data is available, it adds a lot of extra value in terms of tracking change, because it's not always the disappearance of a species that should alert decision makers, but it should be the reduction in the abundance that should be a trigger as well. And then we've put a lot of emphasis on developing visually appealing dashboards and reporting mechanisms. And I've just displayed an example here where we looked at biodiversity data in the Cape Folds freshwater ecoregion of the world. And you see on the right-hand side a multi-site overview dashboard, which gives you a lot of summary statistics and pie charts. And then when you dive deeper, in this case, the SAS data, you get some overview data, geocontext data, which is grabbed for each site. And Tim will unpack that a little bit when he discusses the technical aspect. And then a lot of visualizations. This again was driven by the requirements of the end users, use of color for linking ecological category with charts, photographs, maps, pie charts, and then allowing, in all instances, drilling down to the original data, because obviously you want to be able to see the original data from the summary dashboard. So it's very much a hierarchical drilling down from an overview all the way down to the actual data for the site. Another example, this is a fish species recorded in one of our strategic water source areas, the Boerland. You can see on the right-hand side details of origin, endemism, conservation status. And when you drill down onto the dashboard, you then see the pie charts in more detail, giving origin, endemism, conservation in sampling method where available, and then the biotopes where sampled. And the data is then provided as a time series bar charts. So you can see the earliest record in this instance was in the 1950s, all the way up to the most recent, which is this year, 2021. Split up by non-native and native species in the case of origin. In the case of endemism, all the different endemic levels, as well as conservation status, again, linking to IUCN for this information. And then very importantly, providing the associated metadata that comprises the data that's presented in each of these dashboards. And if you click on any of these DOIs, you will go to the relevant scientific paper, if it was an article or the university theses or the PDF, which is stored on our system. Okay, I think that's really where I want to leave it. I think probably the best is to move straight on to Jeremy and then Tim, and then have a nice open discussion at the end of the session. So I will stop sharing my screen and hand over to Jeremy.